So there's something that's known sort of tongue in cheek as the central dogma, right, of molecular biology, and that is basically the idea that the instructions, right, for sort of building every cell are actually contained within the genetic material in that cell. There's a molecule known as DNA, right? You have all these chromosomes that are composed of genetic material, DNA, but also proteins. And they're kind of the DNA is kind of wound around segments of proteins. These segments, these clumps are known as histones. In individual cells, the DNA kind of unwinds in specific locations. It's a double-stranded molecule, and allows for messenger RNA to form. There's a complementary code, right, between the 4 letters of DNA, and then the complementary nucleotides that sort of sequence based on that code in the messenger RNA, and the formation of that what's called messenger RNA, which is basically transcribing the instructions for ultimately building a protein, building some kind of a protein machine. Is in a segment of DNA that's often referred to as a gene. This process is called transcription. So building the mRNA, the messenger RNA, based upon the code that's present in the DNA, is known as transcription. And then that mRNA leaves the nucleus, and it encounters another kind of machine here. It's made of both protein and nucleic acid. It's called a ribosome. It's a little organ, an organelle within inside a cell. And the mRNA feeds into that, and every 3 letters in the mRNA is linked by what's called a transfer RNA, which is a complementary code, to those 3 letters, to add an amino acid. And so there's like an amino acid sequence that develops based upon the code, in the messenger RNA, which was transcribed initially, the process of transcription, from the unwound segment or gene of DNA. And then that process of, linking up these chains of amino acids to form, the ultimate protein that was coded for by the gene, is known as translation. So the mRNA is now being translated, into the actual protein. And these proteins are remarkable. In other classes, we go into great detail about, receptor proteins, which can receive neurotransmitter messages. There are transporter proteins that can utilize energy in the cell to grab something from one side of a membrane and shove it to the other. There are channel proteins that have holes or gaps that allow for ions and other substances potentially to go through. So these proteins are really remarkable because they build you, right? And proteins can also change the expression of specific genes. So some of the proteins, that are going to be, initially transcribed, the mRNA transcribed, right? And then that mRNA translated into a protein. Well, some of these proteins will then actually bind to certain segments of the chromosomes and switch on entire programs, basically, generate, major changes within inside cells. And during the process of development, this is really important because it kind of can help to set the fate or determine how, different, chromosomal instructions, which chromosomal instructions will actually be switched on or switched off in a cell. But this is the central dogma of molecular biology. Gene expression, right? That's transcription. The reading off of individual segments, to form these messenger RNAs, which then leave the nucleus and then get fed into a ribosome and get translated, basically into an amino acid sequence to build, a specific protein machine. And that's going to help determine, obviously the structure and thus the function